welcome to another episode of BioLogic Science News. Today I want to talk about a newly discovered enzyme that has the potential to revolutionize our use of biofuels. The enzyme in question is one of a rare class that's directly powered by light, making it photosynthetic. Other photosynthetic enzymes are used in plant photosynthesis, but this particular enzyme was found in the cells of the algae Chlorella variabilis NC64A, which belongs to a family of algae known for their glucose-methanolcholine oxidoreductase enzymes that decarboxylate free fatty acids and turn them into N-alkenes or N-alkanes. To do this, the enzymes require blue wavelengths of light. The researchers who made the discovery found that the enzyme has an internal region shaped like a tunnel or a long tube, which is hydrophobic. This means that this region of the protein is repelled by water and other polar molecules, but it attracts and is attracted to nonpolar molecules like lipids. What happens on a molecular level is that the fatty acid is pulled into the tunnel and shuffled down until it comes to a protein structure called a flavin adenine dinucleotide cofactor, or a FAD cofactor, which gets excited by blue light and performs the decarboxylation. To put this phenomenon in layman's terms, the protein tunnel attracts, a fat molecule, and this slides into the tunnel until it comes to the active site of the enzyme, where the important steps of the chemical reaction really start to take place. Here's the working idea that the researchers have for the chemical process. The FAD cofactor gets excited by blue light, which causes it to take an electron from the fatty acid. Stealing this electron destabilizes the fatty acid, and the molecule begins to collapse. This collapse leads to the expulsion of a carbon atom and 2 oxygen atoms from the fatty acid. So to really understand what's going on here, you have to appreciate the chemical structure of the fatty acid. It's composed of a long strand of carbon atoms, which is essentially a hydrocarbon chain. It's what we're trying to get, what we're trying to produce. It's what we use for biofuel. But it also has a pair of oxygen atoms bound to its carbon atom on one end of the chain. The FAD cofactor gets excited by blue light, and it takes an electron from that carbon-oxygen-oxygen complex, and this breaks down and gets expelled as CO2 in the subsequent molecular collapse. The result of removing the CO2 is the creation of an energy-rich, long-chain hydrocarbon, which can be broken down to yield the energy contained in the carbon-carbon bonds. The fuel you pump into your car is composed of molecular hydrocarbons, as is the fuel derived from a typical biofuel process. This process isn't a whole lot different, except for the fact that the enzyme is activated by light. That fact is important, because if the enzyme can be isolated, it can be produced in huge vats, and it can be spread out and just saturated with blue light, turning it into a clean, easy mass production tool for hydrocarbons. Naturally, the devil is in the details, and this process will require a lot of follow-up study and tweaking before it can be used for practical purposes. But it's pretty exciting that we've discovered a way to potentially produce long-chain hydrocarbons with little more than fat and sunlight. And because fat comes from naturally existing compounds that are already in the carbon cycle, this kind of biofuel technology would be carbon neutral. From a more industrial perspective, organic chemists can really have a field day with this FAD molecule. It offers them a lot of opportunities. The FAD cofactor is like a subunit in the protein, and its principal action is to move electrons around. And fundamentally, that's the basis of most chemical reactions. The researchers believe that this particular protein structure with this particular cofactor might be a viable candidate for alteration. It could be altered so that it could perform electron transfer reactions with different compounds, different chemicals. If these efforts are successful, this one enzyme might find 1,000 new applications in hundreds of industries.